Right, can everybody hear me? About 18 months ago, I think it was, I received a letter from an animal welfare charity in the UK inviting me to go along and meet Ray and Jean Greek, where they would be answering questions about their book, Sacred Cows and Golden Geese. Now, I must admit I'd never heard of the Greeks until that time, and I was interested in what they might say in their books. I went and got a copy, and to be honest, I was completely horrified by it, because I think they had completely attempted to rewrite history, and they had clearly failed to understand exactly how medical research is done and how animals are used as models and how alternatives indeed are used as models in the process of biomedical research. So I went along to the meeting and we had a very interesting, free and frank discussion, I think it would be called. And as a result of that, I came back and tried to find whether anybody had written a review of the book. Apparently nobody had, so I sat down and wrote a book. And it was a highly critical review of the book, which I think was justified. Ray, following that, wrote an extensive rebuttal. He wanted it to be published in the same place, Frame News, but as it was twice as long as my book review, Frame said no. And as a result of that, Ray challenged me to a duel, which is why we're here now. This is the modern equivalent of a duel. Okay, so what basically do they say in their book? They say humans and each species of animals differ. Nothing new there. Everybody can agree on that. Therefore, animals can never be scientifically valid models of humans. And this is their basic thesis. Because of the difference between animals and humans, we cannot use animals as models of humans. Animal research, they claim, has never contributed to human welfare, and in doing this, they've rewritten history. The only proper model of humans is humans, and therefore the only proper model of animals in veterinary research is other animals of the same species. So if you want to develop, if you want a drug for dogs, you've got to do research in dogs and so on. Now straight away you can see the weakness if we just consider this last point. Dr. Jean Greek is a vet. Now suppose one day she gets a call from the local zoo. Can you come along and help us? We've got a tiger who's sick. And so she goes along and she sees this tiger and it's got maybe a splinter in its paw. What does she want to do? She wants to give it a tranquilizer so she can then anesthetize it and maybe give it a dose of antibiotics, clean up the paw and so on. What can she do? Nobody has used tigers to develop drugs and anesthetics and tranquilizers for tigers. There are no drugs developed on tigers. So what would she do? Of course she would go and see what drugs and anesthetics and so on are available in other species. Now, I don't say that she could necessarily use them all just as they are, at the same doses used for other species, but with caution, she would almost certainly find that she could treat her tiger with the available drugs available in the veterinary sphere. Okay, so now what we really want to question is, given that humans and animals differ, how can animal research ever be scientifically valid? What I will do is show how and why animals and alternatives can be used as models, even though they do differ indeed from animals. I'm not going to discuss all the major achievements of animal research because there simply isn't time to do that. I will accept that each animal research project must be scientifically and ethically justified because, of course, it is only too easy to do duff experiments. There is no doubt at all about that. I will show that the philosophical justification for the use of animals as models and for the use of alternatives as models is in fact identical. And if we say that we can't use animals as models of humans, philosophically we would have to say also that we can't use any of the alternatives that you are all developing, because after all, a test tube full of even human cells is not the same as a human. Okay, so where do we go from here? Mark Swatowski, an American philosopher, has written a book on models where he says theories, hypotheses, models, and analogies I take all to be species of a genus. And my thesis is best stated directly by characterizing this as representation. So we use animals as representations of humans. He goes on to say, there is an additional trivial truth which may strike some people as shocking. Anything can be a model of anything else. This is to say no more than that between any two things in the universe, there is some property which they both share. So there can be a vast asymmetry between the likenesses and the dislikeness between a model and the thing we're modeling. And I want to use an analogy to explore this in a little bit more detail. My grandchild lives in New York, and last summer we took her to Brooklyn Botanic Gardens. And when you go into Brooklyn Botanic Gardens, they give you a map. Now, a map is a model of Brooklyn Botanic Gardens. Clearly, it is a model, a representation of Brooklyn Botanic Gardens. My 6-year-old granddaughter immediately took control of it, and we had to tell her where we wanted to go, and she showed us how to get there, taking control of the map. Of course, when you come to think of it, children immediately know all about models because we supply them with models as toys. Model dolls, model humans, model trains, and so on. What is the characteristic of this model? How is it similar to the real Brooklyn Botanic Gardens? Well, it's only got one similarity. The spatial orientation or spatial relationship among the features on this map are similar to the real Botanic Gardens. There are millions of differences. Think of Brooklyn Botanic Gardens, which has insects, plants, soil, buildings, and so on. So there are millions of differences. And just because there are millions of differences, that doesn't mean to say it's not a useful model. The other thing is that the scales are different here. This is an important point about models, that models do not, sometimes the differences between the models and the thing we're modeling is very useful. You can imagine a map which was full scale of Brooklyn Botanic Garden, covering several acres, would be totally useless for our particular purposes. Okay, the next point is that maps are useful only for a specific purpose. And this again is the point about our models. We don't use them to model everything in humans. We use each model specifically to model a particular characteristics. And in those circumstances, we can indeed use them for prediction. A model, of course, has to be validated. In the case of this particular map, we would validate it by looking to see whether when we enter the gate at the top left-hand corner, whether we see a long avenue of trees with grass. If we didn't see that, of course, we would begin to think that we had either got the wrong map or we didn't know where we were. And, of course, you must have the right map. If you can have the wrong map, then catastrophe could ensure. Last week, was it, or last month, 8 miners in this country were given the wrong map, as a result of which they nearly lost their lives because they broke through into a tunnel which was flooded. So you need the wrong map. And models can provide new information. I mean, the moment I was given that map, I knew a whole lot about Brooklyn Botanic Gardens without having to go and explore it and find it all myself. Okay, so what we're going to do is look at a couple of models. Ames test. Here is an in vitro model. Chemicals which disrupt DNA may cause mutations and cancer in humans. Bacteria and mammals both have DNA, no doubt about that. Mutagenesis in bacteria is easily tested. Therefore, the bacteria in Ames test can be used as a model of human response to chemicals which may or may not cause cancer, may or may not be carcinogens. So the Ames test has been validated to some extent. It's not 100% valid. Its accuracy is in some doubt, I must admit, but it's still regarded as quite a useful test. As an animal model, we'll look at the rabbit response to an injection of insulin. Insulin reduces blood glucose in both rabbits and humans. Therefore, rabbits can be used to model humans in testing the potency of insulin preparations. There is no doubt about that. That's a predictive model. It has been valid repeatedly because it's been used for more than 60 years in testing the potency of insulin preparations. It's no longer a necessary model because non-animal assays have now been developed. But it is still a scientifically valid model. Okay, what about animal models in the development of drugs and vaccines? Well, just three very quick examples. Anti-rabies vaccine developed in about 1885 by Pasteur using dogs and rabbits. The point about the use of the dog here is that rabies is a virus which affects both humans and animals, and therefore animals could be a model for developing a vaccine. And in fact, having developed a vaccine against rabies, which was effective in dogs, Pasteur found that the same vaccine was equally effective in humans. Salvarsan for treating syphilis. Ehrlich screened more than 600 arsenic compounds in rabbits. He was able to do this because a colleague had developed and discovered that rabbits could be infected with syphilis. So he was able to screen a large number of compounds to see whether they were effective against syphilis, and he found one of them, salvarsan, which was. And indeed, this sort of process has been used repeatedly in drug development. Of course, one needs, following the discovery of a drug which will cure the disease in rabbits, of course one needs clinical trials. That stands to reason. One needs clinical trials, and some of them will fail. Nevertheless, it is one way of developing drugs, and so far no better way really has been developed, although maybe in the future we will develop better ways. Penicillin was isolated in 1929 by Fleming. He couldn't isolate it. Chain and Florey did so. And this is a good example of the use of an in vitro model because, of course, penicillin will kill bacteria in vitro in a test tube, and therefore they didn't need to use an in vivo model in the isolation of penicillin, but they did need to use it in testing the efficacy and whether it was toxic, and in that case they used mice. So we now have a whole lot of vaccines that have to be tested still in animals. And until such a time as we have alternatives, we will have to continue to use these animals to test the safety of these vaccines. For example, polio is a live vaccine, and it's notorious for reverting to neurovirulence. If we did not test it in animals, then every so often we would give a batch of polio vaccine to children who would then get polio. And as a result of that, people, of course, would stop vaccinating, they would lose confidence in the vaccination process, and polio would come back as a serious disease. Now, I'll give you an example of a model developed for a very specific purpose at Leicester. A young surgeon working with terminal liver cancer patients. They have tumors in their liver. You can't excise those surgically, but what he has is a new probe about the size of a pencil which he can put into the tumor and give a dose of microwaves and cook it. So the question he asked is what dose of microwaves would be needed to cook a tumor of a given size, and does cooking a tumor sitting in your liver harm the patient? Now, he couldn't test this on patients because really it would not be at all ethical to do so. The models he used was pig's liver from a butcher. He tried this first, but of course pig's liver from a butcher is not the same as pig's liver or human liver, live liver, because there's considerable blood flow through liver. So he used 13 pigs, giving various dose levels. The lesion diameter was recorded, and the fate of the lesion was recorded. After a couple of months, it had shrunk away, and he was then able to treat so far 9 terminally ill patients who were expected to die, and as a result of this procedure, they are now living. So, okay, but Christopher Columbus didn't have a map. Sometimes we need to explore things a little bit. The procedure is quite straightforward. We study another life form because it's easy, practical, and often ethical. We find a phenomenon that likely to be of medical importance. We say, is it also seen in humans? If it is, we've got new knowledge. If it's not, then we don't have new knowledge. So I'll just very quickly give some examples. Mendels' laws were developed using peas. Now, Mendel was not aware that he was developing a model for the mode of inheritance of cystic fibrosis in humans. So in that sense, it was an orphan model. He'd developed the model, but he didn't quite know what it was a model of. Muller, in 1927, discovered that irradiation caused mutations using Drosophila. There are some other examples there which I think I'm going to skip over because I'm running out of time. I'll just come to this last model here, human obesity. This is a major problem. Here is a genetically obese mouse. We don't know how appetite is controlled, and this mouse maybe gives us an opportunity to learn that. It's been around for about 40 years. Recently it was cloned. We didn't think it was a model of any human disease because, of course, human obesity we thought was never controlled by a single gene. But lo and behold, once we cloned it and discovered that it coded for a protein called leptin, a human family with this particular deficiency was discovered. So straight away as an animal model and a human equivalent. The same mutation, as I say, is found in humans. And now this gives a stimulus to explore what are the genes that control appetite and how might appetite be controlled. And you might have seen that was a paper last week in Nature in which the hormone PYY336 which is secreted in the intestines, was given to mice, rats, and humans in a controlled double-blind trial, and it was shown to reduce appetite very substantially. So let me just finally add this, which is not strictly relevant, but it is important to remember that if you live an average lifetime of 75 years in the UK, we've got very good statistics on numbers of animals used, your total usage of animals will be about 4. You will use, as it were, or other people will use on your behalf, 2.4 mice, 0.4 of a fish, 0.8 of a rat, and 0.4 of other animals. So that is a total requirement, as it were, for you for a lifetime medical research. So I will conclude here. Yes, we still need animals in medical research until we can develop alternatives. Animal and alternative models have the same philosophical justification. They do not need to be like humans in every respect. They do, of course, need to be validated. Models can provide new information. I might come back to the 747 model a little bit later. Scientific validity alone, of course, does not justify use. Each project needs separate and scientific and ethical justification. Thank you.